Hello and welcome to biology class. We are studying topic biochemistry of cell. And so far we studied about carbohydrates, another biomolecule that is proteins, we have studied about lipids, and lastly we reached to the study of nucleic acids. And in the last two videos we studied many things about nucleic acids. Now few important things which we have studied, remember, and which are important to understand this particular video. First thing is that nucleic acids are also polymers which are made up of a monomer called as nucleotides. Okay, in this video we are going to learn little bit more things about nucleotides and their joining. Let us go ahead and study that. Then another thing you need to remember about the nucleotide is that nucleotide is made up of three components. One is phosphate group, another one is pentose sugar, and another one is nitrogenous base. Now here I am not talking about a nucleoside. Nucleoside is one thing which you need to remember. It is not applicable here in this topic. It will come in another topic next year. But right now we are just talking about nucleotide, which is nothing but all the three components: phosphate group, pentose sugar, and nitrogenous base. Remember this. Now these nucleotides are monomers of nucleic acids. What are those? Monomers. Those are monomers. Now these monomers basically give rise to two different type of nucleic acids. Monomers, that is nucleotides, give rise to two different type of nucleic acids. One type of nucleic acids are called as RNA. This is the structure of RNA. Let me take it complete diagram. So one is RNA and another one is DNA. So basically we are going to deal with only two different type of nucleic acid. One is RNA and another is DNA. So taking into account both DNA and RNA, I will explain you how exactly these nucleotides join to one another to give rise to both nucleic acid. How it gives rise to DNA and how it gives rise to RNA. So let us start studying it. But before starting to joining these monomers, that is nucleotides, we need to understand few more very important things about nucleotide. So let us revise those things. I taught you them, but still just take a quick revision so it is easy for you to understand DNA and RNA and how exactly DNA is formed and how exactly RNA is formed. So let us begin. I'm going little faster, just it is a quick revision, that's why. Okay, so phosphate group, first component, sugar, second component, and nitrogenous base, third component. When I talk about phosphate group, in both DNA and RNA, phosphate group is same. There is no difference in the phosphate group. There is a difference in the pentose sugar. In RNA it is ribose. In DNA it is deoxyribose, correct? Deoxyribose. That's why named DNA, correct? Now let us talk more about nitrogenous bases. Now when I consider nitrogenous bases, there are two types of nitrogenous bases. I am just writing short form. One is purine and another one is pyrimidine. Which two are purines? A and G. Single ring or double ring? Purine. Simple ring, simple name, complex structure. So double ring. Pyrimidine, C-U-T, cut is the pyrimidine, correct? Cut pi. So when we consider these three are pyrimidine and these two are purine. Pyrimidine, single ring structure. Okay. Now when we consider DNA and RNA contain both the purines, that is it contain A, G, but in the pyrimidine it contains C and U. See, it contains two purines, A and G, and C and U are the pyrimidines it contains. Got it? In the DNA, A and G is there. A and G is there. And in the pyrimidine, it is having C and T. Remember this thing? DNA never contains uracil. Uracil is a specificity of RNA. And DNA contains thymine, which is never present in the RNA. Okay, so it's simple to remember. In the DNA, U is replaced by T, and in the RNA, T is replaced by U. Okay, basically, when we talk chemical structure, uracil and thymine are very close to each other in their chemical constitution, but still RNA never contain thymine and DNA never contain uracil. Okay, remember this. Okay, now further things about purine and pyrimidine I will explain you while joining them together. Understanding? So is it clear? I'm revising once again. Phosphate, in both DNA and RNA, phosphate is same. Okay, pentose sugar, RNA is having ribose sugar and DNA is having deoxyribose sugar. When I talk about purine and pyrimidine, both the purines A and G present in both of them. DNA also contain A and G, as well as RNA also contain A and G. But in the pyrimidine, RNA contain C and U, and DNA contain C and T. Got it? Now they are having affinity. Purine and pyrimidines are having affinity among them, and that we will study little bit later on. So it is clear about it? Okay. Now let us try to understand how exactly these nucleotides join to one another to form DNA and RNA. Okay. Now remember this thing, nucleotides need to give two different type of molecules. Now look at the structure of RNA. It is a straight, long, straight and long, and single strand is present. Okay, single strand of nucleotides are present. Means if I am having nucleotides, I join them one after the another, I can make up RNA. Got it? See, if I am having nucleotide one after the another, if I keep on joining them to form a long chain, I can form RNA. But DNA is not a simple structure. You see, DNA is having first of all two strands. They are arranged so differently. We are going to study structure of DNA in details, but right now just try to understand they are having such a twisted structure and two strands are joined to one another. So just like RNA, I cannot make DNA. I need to, follow another method of joining these nucleotides to make up DNA. So let us try to understand. Now, understand one thing properly. There are two ways. See, there are two ways to join nucleotides. Only one way we apply while making RNA, and both the ways we need to join to make up DNA. So let us try to understand how exactly these nucleotides join to one another to form RNA first, and then we will see how exactly they join to form DNA. So let us take a long chain of nucleotides. Okay, I'll take the long chain of nucleotide first, and then I will show you how exactly they join. So here I have taken some few nucleotides, okay, and then now we will try to join them together and make RNA out of it. So let us try whether it is possible for us or not. Let us see. Now see, to join these nucleotides, few things we need to do first. Now I'll do the simplest one first. I'll number all these nucleotides. So this is the first nucleotide. This one will be the second, this is the third one, this is the fourth, and this is the fifth one. So I have taken 5 nucleotides and I arranged them one after the other. Got it? Now, one more thing we need to do to understand the joining properly. We need to number carbon. Okay? Do you know which is the first carbon in this nucleotide? Which is the first carbon? This will be the oxygen. Now tell me which is the first carbon? Clockwise to the oxygen. So this will be the first one, this will be the second one, this will be the third one, fourth, and here somewhere there will be the fifth carbon. Got it? So likewise here also, but I am not writing over here. You just understand it. Now what happened, try to understand. This third carbon will form a bond between carbon and phosphorus or phosphate group of the next nucleotide. See, carbon from the first nucleotide, which is on the third position, it form bond and join to the phosphate of the second nucleotide, next nucleotide. During this process, during the formation of bond, water molecule is removed. So, bond is synthesized by removal of water molecule. What does these bonds are called as? Yes, tell me. Or what does this process is called as? The process is called as dehydration synthesis. So, a new bond is synthesized by the removal of water molecule. The process is dehydration synthesis. Okay, and here the name of the bond which is synthesized over here, it is called as a phosphodiester. What this bond is called as? Phosphodiester bond, or even called as phosphodiester linkage. But the process is dehydration synthesis. What happened? Third bond is formed between third carbon of first nucleotide and phosphate of the second nucleotide. Similarly, there is a joining of second and third nucleotide by same bond. Third carbon of second nucleotide join to the or form bond with the phosphate of the next nucleotide. Likewise, this will also join. Likewise, this will also join. Understanding? So, try to understand, all the 5 nucleotides got joined to one another by bond of phosphodiester. The bond is called as phosphodiester bond. Never forget it. Remember, the first bond which I taught you while formation of nucleotide is phosphoester bond, and this one is phosphodiester bond. Okay, so likewise all these nucleotides will join to one another to form a long chain. Now RNA is formed like this. Now if you see the structure of RNA, this RNA, whatever you see over here, this grey colored structure is nothing but sugar and phosphate join. So here there will be phosphate, agree? And here there will be sugar. Then next phosphate, next sugar, again phosphate, sugar. So here also likewise, here there will be sugar, in twisting there will be phosphate, okay, here somewhere phosphate, okay, then there will be phosphate, then sugar, then phosphate, then sugar. Likewise whole this long chain will be formed. Okay, got it? So this grey colored structure, whole structure is formed by sugar and phosphate. Let us come back to our diagram. Now see, there will be alternate arrangement of phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay, and this phosphate sugar thing remain outside, and inside all nitrogenous bases. In this diagram also you can easily see nitrogenous bases remain inside. These are nitrogenous bases. This stick like structure, no, they are nitrogenous bases. They remain inside and sugar phosphate remain outside. So sugar phosphate, here it is called as a sugar phosphate backbone. It is like a backbone of a person. Okay, they remain outside and protect. That's why it is called as sugar phosphate backbone. So joining of the phosphodiester bond, nucleotides form the sugar phosphate backbone and they keep these nitrogenous bases inside. Okay, this is the first way of joining nucleotides to one another to form a RNA. Clear about it? Clear about it? Only one type of bond is involved in this joining, that is phosphodiester bond. Clear about it? So we'll go to the second linkage, second bonding. Very good. So for this second bonding I need to take some more nucleotides. So I'll just take it and we'll come back. Now, when we consider the second way of joining the nucleotide, it is something different. Now see, the second way of joining is mainly utilized for the formation of DNA. So what happened? Try to understand. First of all, by normal phosphodiester linkage, a one linear long chain of DNA is formed. Okay, so in this, nucleotides are placed straight like this, suppose. Okay, nucleotides are straight like this. Up is the phosphate, middle is the sugar, and here somewhere is the nitrogenous base. Got it? Now I want to join another strand of the nucleotides into the opposite manner. So suppose one nucleotide is like this, I want to join the another nucleotide like this, inverted. Exactly, it was like this, I need to rotate it, I need to invert it, and I want to join to the first nucleotide. See, in the one strand nucleotide is like this, straight. The second nucleotide, to join the second strand, okay, what I will do is, I will take nucleotide, I will rotate it in the opposite manner, and I will join it to the first nucleotide. Got it? Understanding? So let us take it, let us do it. So I'll just take one nucleotide for your understanding, and I'll explain you. So I have taken this one nucleotide. Now I want to join this first nucleotide with this nucleotide. But this joining like this is not possible. I cannot join it like this. The bond is not formed. Bond is not formed like this. Okay, joining is not possible. So what I will do is, I will rotate this whole nucleotide. Rotate in what way? Just try to understand. I'll just rotate and I'll show you. So this nucleotide was like this, I made it like this. Okay, so let us try to join it. So this is the first nucleotide I have taken. I'll just align it in front of the another strand. Now they both are in the straight line. So let me take few more nucleotides and I will explain you how exactly they join to one another. So I have taken few more nucleotides and I'll show you now how they get joined to one another. Now this also need to make one strand first. So now let me explain you numbering of the carbon molecule, and then you will understand how exactly they get inverted. Now see, this is the O. So this will be the first carbon, this will be the second, this will be the third one, this will be the fourth one, and here somewhere fifth one. Okay. Now in this also it will be the first, second, third, fourth, and fifth one will be somewhere here. So first of all, there will be formation of bond between third carbon and phosphorus. So all these bonds are also joined to one another exactly like this previous, just in the inverted fashion. Got it? Just in the inverted fashion. Now in front of one another, here there is a nitrogenous bases, and they are arranged in such a way that purine will come in front of pyrimidine. See, purine come in front of pyrimidine. Now remember this thing, purines are always having attraction towards pyrimidine. These purines are always having attraction towards pyrimidine. Specifically, purine adenine is having attraction towards pyrimidine thymine. Okay, and guanine is having attraction towards cytosine. So when, suppose here I considered it is A should be double ring, I will draw double rings. Okay, then automatically here T will come because they are so attracted towards one another. Wherever A come, in front of it automatically T will come. Okay, suppose I considered here there is a C, obviously single ring structure, then automatically here there will come G, double ring structure. Okay, here suppose I have taken, it is coming suppose a T, then automatically here it will come A. They are having very strong attraction towards one another. Okay, and here let us take again a T and A. Okay, got it? Now try to understand joining between them. See, wherever A, automatically in front of that T will come. wherever it will come C, automatically G will come, and wherever there will be T, A will come in front of that. So A and T join to each other by 2 hydrogen bonds. How they join? 2 hydrogen bonds. And C and G join to one another by 3 hydrogen bonds. A and T again, how many hydrogen bonds? 2 hydrogen bonds. A and T, 2 hydrogen bond, and C and G will be 3 hydrogen bonds. Understanding? Now try to understand what exactly happened. See, we were having one strand constructed, okay, just like joining nucleotides by phosphodiester bond. Got it? Now, as it is a DNA, it should have a second strand in front of it. So there should be a second way of joining nucleotides, because 2 strands should be in front of one another. See this structure. You can see one strand of DNA and in front of it another strand of DNA. Got it? So 2 strands should join to one another. Agree? And I am explaining you how exactly they join to one another. One strand is constructed first, as I explained you in RNA. Just U is replaced by T over here, otherwise the same as RNA. Okay, and in front of that another strand should be constructed. So that is the second way of joining. So 2 nucleotides, suppose one nucleotide is like this, another nucleotide cannot come and join to this like this. It is not a proper joining. It doesn't take place. Okay, suppose this nucleotide is straight, the nucleotide which is in front of it cannot be straight. They cannot join to one another like this. Okay, they should. So what happened? This nucleotide is straight, which is in the first strand. So another nucleotide come and get inverted and get joined to the first strand. Understanding? So what happened? This nucleotide is straight, so whatever nucleotide come in front of it, if you see, it is exactly the inverted one. Got it? And it will join to the first nucleotide. So here joining is between the nitrogenous bases. Whenever nitrogenous base A is there, purine A is there, it is joining to the pyrimidine T by 2 hydrogen bonds. Okay, then what will happen? Phosphodiester bond is synthesized and next nucleotide will come. So this is the first one here and this will be the second one will come. Second one will again go inverted. Okay, and it will join to the first nucleotide by phosphodiester bond. This bond, pay attention properly, and it will join to the first strand, second nucleotide of the first strand by hydrogen bonding. Here suppose C is there, then G will come and G will join by 3 hydrogen bonds. Okay, it is not difficult, don't worry about it. It is very simple. Just you are studying it for the first time, that's why you are finding it little difficult, but it is not at all difficult. It is very interesting, and after few days you will like it like anything. Okay, so is it clear? Then third nucleotide will come in the second strand. See, this third nucleotide is coming and it will join to the third nucleotide of first strand by hydrogen bonding. Okay, these are hydrogen bonding, and here in the same strand it will join by phosphodiester bond. Clear about it? Let me show you with the help of another diagram. So it will be very, very clear to you. Now this is the diagram. Let me enlarge it little bit so it is easy for you to understand. Now this is that structure. See. Now this is, I am having the first strand. First nucleotide. Got it? This is the first nucleotide. This will be the second. This will be the third one. This will be the fourth one, and this will be the fifth one. Now what happened? Try to understand. This is the first nucleotide. It was already attached to one another by these bonds, blue colored bond by phosphodiester bond. Agree? Now, when the second strand should be constructed, that time the first nucleotide of second strand, this one, 1, 2, 3, 4, 5. Okay, it will come inverted. So this is inverted. It will come inverted. It will join to the first strand. Okay, it will join to the first strand with hydrogen bond. See, hydrogen bond, got it. Then the second nucleotide of second strand will come. Now it need to join to the first strand even as well as to this strand. So it will join to the first strand with these hydrogen bondings and to the second strand with phosphodiester linkage. Getting my point? Understanding? Clear? You join by yourself, then you will understand. Draw one nucleotide, in front of that draw inverted nucleotide and get joining one another. Practice it, it is very simple. Okay, now so two strands join one another by hydrogen bondings and the nucleotides of one strand join to one another by phosphodiester linkage. Clear about it? Clear about it? More I explain, more confused you will get. So I am not explaining anymore. So just try to understand it properly. See now, here you will understand. Now when we consider this DNA, you can see two structures of DNA. You can see two strands of DNA. See, detailed structure of DNA I am going to explain you in the next video. But before that, just try to understand what is here. See here, whatever grey colored structure you see, that is nothing but the phosphate sugar backbone. Okay, and whatever you see inside these rugs, these are nothing but nitrogenous bases. Here it is A, here it is T, here it is C, here it is G. Likewise, they are attached to one another with the help of hydrogen bonds. So sugar and phosphate remained on the both side, and in the middle there are nitrogenous bases and hydrogen bonding. Got it? So let me revise quickly so you will understand it quickly and clearly. Now see, nucleotides are monomers. They have to join to one another to form a polymer. Now two types of polymers are there. One is RNA and another is DNA. To form these two types of polymers, there are two ways of joining these nucleotides. One way is by linear manner, in which one nucleotide joined to the next one like this in a linear fashion, in the straight line fashion. And this joining is by phosphodiester bond between third carbon of first nucleotide and phosphate group of the next one. Got it? Then secondly, the second joining is between the two strands. Okay, two strands in which DNA join to one another, because DNA is a double stranded structure. Okay, now to join the two strands together, first of all you need to understand about the purine and pyrimidine linkage. A is always joined to the T and C is always joined to the G. Okay, C and G join to one another with the help of hydrogen bonds. Okay, now these hydrogen bonds, three hydrogen bonds formed between C and G, and two hydrogen bonds are formed between A and T. Got it? Clear about it? Now next thing, so this sugar and like when the joining of DNA takes place, that time one strand is straight and another strand is nucleotides come inverted and they join to one another. So both the types of bonds are formed. One is hydrogen bond between the two strands and another one is the phosphodiester bond between the same strands. Got it? Clear about it? Any more confusion? If you are having any confusion, draw it by yourself. You will clearly understand it and quickly understand it. Draw a nucleotide and try to join it to the another nucleotide. And specifically give number to the carbons, so it will be very clear to you. So pause the video over here and then draw the joinings, draw the nucleotides and try to join them together. Now after this joining, you need to understand only one more thing that I am going to tell you, and then in the next video we will study structure of DNA. Now after understanding this, understanding structure of DNA will be quite easy for you. So now let me tell you only one thing and then we'll get over with this video. See, this is the first strand, correct? And this is the second strand. These two strands are joined to one another by hydrogen bonding. Okay, now let us try to understand how exactly we give prime to this strand. Now see, let us consider carbon number. I am telling you again and again, understanding carbon number is extremely important in this particular understanding structure of nucleic acids. So this is O, remember, this is O. This is the first carbon, this is the second one, this is the third one, this is the fourth one, and this is the fifth one. Agree? Okay, I'm just explaining you one thing. Now phosphate group is always attached to the fifth carbon of sugar. Agree? Phosphate group is always attached to the fifth carbon. So this is called as 5' end. This strand, first strand, it is called as 5' end. Now let us consider the last nucleotide of this strand. Now again, this is O, this is the first carbon, this will be the second one, this will be the third one, fourth, and this will be the fifth one. Agree? So this strand starts at 5 carbon because it starts with phosphate group and phosphate group attached to the fifth carbon. And this strands end at the third carbon, okay, because this is the third carbon of the which is free over here. The phosphate group is not attached. So this is the third carbon. So the strands start at 5' and end at 3'. So the running of this first strand is 5' to 3'. On the other hand, see the second strand which is in front of it. Let us mark the carbon O, first carbon, second carbon, third carbon, fourth and fifth carbon. Okay, so this strand starts with third carbon. Okay, and end at, let us see the last nucleotide of this carbon O, 1, 2, 3, 4 and 5. So it is ending at the 5'. So this dash, never say 3 dash, 5 dash, it is a wrong method. It's said as a 3', 5'. So in every DNA, one strand run from 5' end to 3' end and another run from 3' to 5' in this direction. The significance of it will come when the DNA is formed. In the next video, I'll teach you the structure of DNA. Till then, quickly we'll take a revision of what we have studied in this video. Nucleotides join to one another to form nucleic acids. One is RNA and one is DNA. There are two ways of joining of these nucleotides. One is in the linear fashion, which takes place in the RNA as well as in the DNA. Another fashion is the joining two strands of nucleotides together. It is by the hydrogen bonding. Okay. So with this knowledge, we'll end this video over here. In the next video, we will study structure of DNA. Till then, goodbye. Take care.